And today, our speaker is Dr. Liana Rosenthal. Now, I did ask for a bio, although I kind of did it last minute, and she just sent it a few minutes ago. But in reality, I'm just going to make one up. So Dr. Rosenthal was Miss Mississippi in 1992. She was the winner by far. And then after her short stint as a beauty queen, was an auto mechanic for Dale Earnhardt's race car team, and then went to med school. And now she's the director of the Ataxia Clinic at Johns Hopkins. So Johns Hopkins Ataxia Clinic, she's the director. So she's a pretty smart cookie. That part of it is true.
Very talented.
Well, thank you very much for having me. And thank you for the very warm introduction. And I, of course, it's all 100% true, clearly. So way back when Dr. Snyder invited me to come speak today, the thought was that I was going to be talking about the COVID vaccine. And a lot has happened since she last, asked me to come speak. And so at this point, there's been a lot of, at this point, I think they say that almost every adult who wants the COVID vaccine can get the COVID vaccine at this point, which obviously was not true, 3 months ago and 3 months before that. So what I decided to do is and with sort of Linda's advice, is that we're going to talk about research in general. And I threw in there some information about the COVID vaccine in the context of talking about research. And then we're also going to talk about some ataxia research, both current ongoing things and some ataxia research that's coming down the pipeline. So the idea is to sort of give an overview of research, talk about COVID, and then the COVID vaccine, and then talk about other ataxia specific research trials that are ongoing. So with that in mind, let me see if I can share my screen here. And, despite having done this numerous times, it's the whole sharing and having it still look pretty. So here's sharing. Now I'm going to, if I do this, what do you guys say? Do you guys see the full slides, or do you see the does it look like it's in speaker mode?
Presentation mode.
It's in presentation mode.
Hold on, how's that? Is that better?
That's better.
Perfect. All right, good. So if people were glancing at that quickly in the presentation mode, they would have seen that there was 69 slides. There are not actually 69 slides. There's actually about 30. Because I know whenever I see anyone with slides, the first thing I look to see is how many of these am I going to have to sit through. There's only actually about 30. Yes. It's just an ongoing side sets that get moved around. With that in mind, please jump in if I am saying things that don't make sense to you, if I'm being confusing, any of those things, please jump in. The whole idea is to make it understandable and informative and interesting. So please interrupt. I can't see all of you because you know how Zoom is. So I'm not able to actually see all of you guys. So if someone has a question, please just unmute yourself and interrupt. It is not a problem, okay? All right. So we are going to be talking today about types and categories of research. We're going to be talking about how a molecule becomes an FDA-approved medication. We're going to talk about the COVID vaccine research and myths. And then we're going to be talking about ataxia clinical research. And I tried to put some cartoons in there to make it kind of fun. And so I like this one that, under strict laboratory conditions, research concludes that in spite of being watched, pots do eventually boil. So, all right, so types and categories of research and how a molecule becomes an FDA-approved medication. So first, there's basic science research. And the idea of basic science research is investigation of a subject to increase our knowledge and understanding about it. It's knowledge for knowledge's sake. We want to learn more about the world, and the world is an interesting place, and so we want to learn more about it. In the sciences, basic science research usually refers to things performed on cells and animals. So we talk about petri dishes, we talk about animal models, mouse models, rat models, that kind of thing. So it is knowledge for the sake of knowledge, but it can then be translated into other things. Because what we find time and again is that this basic science is increasing our knowledge about whatever it may be, actually in turn can improve our lives or our health in some other ways. So an example is someone created and looked into and investigated an atomic clock, and that is now critical to our satellite-based GPS. So every single time we, go on our phones and try to figure out where we're going based on GPS, that was originally that information, that science that allows us to do that was originally basic science that then got translated into something that makes all of our lives better, because now we can figure out where we're going based on GPS. So that's basic science research. Rats, mouse, mice, that kind of thing, cells, petri dish. Clinical research is any research that involves humans. And the goal is to improve human health or increase our understanding of human biology. And clinical research may be observational or interventional. So observational clinical research is literally we observe. And we use that word observe kind of loosely here because we observe clinical assessments, but we also observe blood and spinal fluid and imaging and things like that. So I use observation and observe kind of broadly. It's observational research. Interventional research, those are clinical trials. We determine if our intervention changes an underlying condition. So in a clinical trial, there is often one group that gets the intervention, one group that doesn't get the intervention, they get the placebo. And the question is, does the group that gets the intervention, do they do any better than the group that didn't? So for examples, observational studies, some well-known observational studies are the Framingham Heart Study. The US Census is a well-known observational study because you count the numbers, you're observing the population. And then COVID vaccine monitoring, observational trials. So not the COVID vaccine itself, which I'll get to, but the monitoring. So the studies that have now come out that said, there's one that came out just this past week that said of 6,000 doctors vaccinated in Israel, of doctors vaccinated in Israel, 5,800 or something were vaccinated and 200 weren't. And of the 5,800 that were vaccinated, there was, this many of them got COVID and this many didn't. That is an observational trial because they are observing. It is not up to the people running the trial who got the vaccine and who didn't. It was simply observing. And I like this cartoon because it kind of speaks to a lot of the medical news we get, less so on COVID, but it's sort of, coffee, something can cause something in something. So coffee can cause depression in twins, or you could put it somewhere else. Smoking can cause hyperthermia in men 25 to 40. So I like this cartoon, according to a report released today. So these are all observational studies. Then there's clinical trials. Every prescription drug or vaccine you have ever taken was once part of a clinical trial. They determined, do people who get this drug, this vaccine, do better than people who don't? So antibiotics, birth control pills, blood pressure medications. I listed here some medications commonly used in people with ataxia, so riluzole for immunopurity and amantadine, baclofen, all part of clinical trials. And of course, the COVID-19 vaccine, all part of a clinical trial. So far we've talked about basic science, clinical research. In clinical research, we've talked about observational studies and clinical trials. So those are kind of the big categories. And often what happens is the basic science leads into the translational and clinical research because it flows together. We have to look at a molecule in a petri dish and look at a molecule in a mouse model or a rat model and see how that molecule does. That's the basic science. And then from there, it goes into the clinical research and the clinical part of it to see how that molecule does in humans. So how a molecule becomes an FDA approved medication, usually about 12 to 15 years, sometimes more, often more than $1 billion, much slower than any of us would like it to be. But here's an important point. It clearly can be accelerated in an emergency and with lots of money. And so I think this is where the COVID vaccine comes in. And I'll get to some of the interesting background on the mRNA vaccines, the Pfizer vaccine and the Moderna vaccine. I'll get to some of the background about it. That really, it took more than 12 or 15 years, but that final push, that final acceleration was basically in an emergency and with lots of money, and they got rid of all the red tape. That's how they did it. So I like the cartoon about how, after reading about all about cancer treatments, trials, phases, and stages, I've decided to quit reading. It was just too much, too overwhelming. And so these are the stages. Usually there's lab studies for several years, and that's considered preclinical. Then there's phase 1, which is usually, tens of people, first in human kind of thing, or phase 1. Then phase 2 are expanded safety studies. Is this safe? Is this drug that we're giving safe? Phase 3 is this drug that we're giving effective? And then what they don't include here on this cartoon is there's a phase 4, which is monitoring. So it's after phase 3 that things go through the FDA approval process. If they get approved, then phase 4 is the subsequent monitoring. And then this was a nice, very pretty visual, but I also liked this one because it included more of phase 4 and it included more of the years. So they're saying 6.5 years in preclinical, 5,000 compounds evaluated, 5 enter trials, 20 to 100 people in phase 1, 100 to 500 in phase 2, 1,000 to 5,000 in phase 3. And then you're talking FDA approval. And if you notice here, they claim 1.5 years for FDA approval, but clearly that process can be shortened, right? I would point out, and it comes up a fair amount in ataxia, and for people who are thinking really critically about this, if a phase 3 study normally has 1,000 to 5,000 people, there's just no way that can be done in the ataxias. It's just not possible. We don't have, finding 5,000 people with SCA3, it just doesn't exist that we can do that. So often what happens for these rarer drugs is there's different ways of doing it. The phases are the same, but we're allowed to get away with smaller numbers because if you treat 5,000 people with SCA, you're not going to have anyone left when the drug is approved. Yes, I see his question. Go ahead.
So with a lot of the ataxia clinical research, they want you to know what type of ataxia you have. They want to do research on SCA1 or 3 or 6 or whatever. And they're very specific about it, but a lot of people have not undergone the genetic testing to find out what they have exactly. 1, because it's expensive, and 2, because a lot of doctors where they live maybe aren't familiar with how to go about ordering it or things like that. And that I think that was the problem in Omaha for a long time, is that there was not an ataxia specialist here. And so people weren't getting tested to know exactly what they had. We have an ataxia specialist now and a genetic counselor to help with that piece, but 10 years ago we didn't.
We actually have a new research clinic at the university here now.
So what do you recommend for people that haven't done that in the past?
So I think there's a couple of different ways you can do it. There are a lot of these clinical trials and a lot of these pharmaceutical companies, if they are recruiting patients with SCA3 or SCA1 or 2 or whatever, they will pay for you to be tested. If you are interested in their research, and only if you're interested in their research, they will actually pay for you to get tested for whatever SCA they want in their trial. So that's one way of doing it for free. But if you're going to do that, that means you really are genuinely interested in doing that clinical trial. So you just have to be careful with that, because sure, it's great to get free testing, but then you also are sort of agreeing to be part of that clinical trial, which you may or may not want to be, depending on the details. I would say a couple things. The cost is a huge issue. And I think I will talk a little bit later, but the National Ataxia Foundation actually has created a pre-competitive drug development group of 10 pharmaceutical companies. And one of the things they're looking into is ways of paying for genetic testing in the US to try to basically increase the number of people eligible for their studies. But what they would then be doing is they'd be paying for people to get 1, 2, 3, 6, 7, 8, and 10 for the most part, because those are the more common ones. And so, unfortunately, that's the situation. Ultimately, there's the cost, and then there's the counseling and the access issues. For the cost issues, talk with your insurance, talk with your genetic counselor, talk with, and I believe you guys have something as well, don't you, for the Nebraska ataxia folks? Yeah, you guys have which most places do not have what you guys have, where there's someone who can help pay. That's actually huge.
Yeah, if you apply for a grant for that, we will help cover that cost.
Which is amazing.
It's a Nebraska, Iowa benefit.
Okay.
If you live in Massachusetts, it doesn't help you.
No, it's not going to help you. And that's what you guys have is amazing, and what you've set up is amazing. We, as in the National Ataxia Foundation, physicians everywhere, are well aware of costs as being a huge barrier to getting tested. And it's one of the things that the pre-competitive consortium at the NAF is actually working on. And is there a way that we can do a centralized genetic testing and pay for at least the more common ataxias? The idea being that we want to test people, we want to test for the ataxias for people that we're about to come up with disease modifying therapies for. So, it's wonderful to have a diagnosis of SCA 23 because it's an answer. That's a really important thing. But in terms of, how much money exists in the world and time, right now we're prioritizing testing the ones that there's going to be a drug for soon. Because, if you have SCA3, but you don't know you have SCA3, and there's now suddenly a drug for SCA3, then you're really missing out on that opportunity. You basically, for the people that don't even have access, unfortunately, those are the people that aren't even on this call, right? And so finding those people is actually really hard. And, we're all trying, but even on just yesterday in clinic, I met with a couple that lives in Florida whose physician told them, 6 months ago, 3 months ago, maybe even, yes, you have ataxia. There's nothing more to do. No more testing, nothing. The patients never had genetic testing, never had anything. And so they managed to find the Hopkins Ataxia Center because of some videos we put out about, exercise and ataxia. So they discovered that it existed, basically. But, I think it's really hard getting that word out. One of the things I've looked into and thought about are various continuing medical education things that we can do to try to educate physicians more. But really, it's hard. Am I answering your question? I'm kind of rambling. Does that answer your question?
You're absolutely, yeah.
So here is some other clinical trial and research terms to know. So placebo is a substance or thing with no therapeutic effect. So these are the placebo-controlled trials. So, half the group gets the placebo, the substance or thing with no effect, and half the group gets the drug. So in the Pfizer vaccine trial, I think it was 44,000 people, 30,000 of them got the vaccine and 15,000 got placebo. It's something numbers like that. Double blind means the researchers and the patients don't know if they're taking placebo or active medication or intervention. The patients don't know, the researchers don't know. There is a code, so someone somewhere knows, but the doctor's doing the evaluation and the patients themselves don't know. Sometimes we'll use the word powered, and we say a study is or is not powered. And the idea is the ability of a study to detect a difference, if a difference actually exists. Detect a difference in the placebo versus the active substance if it actually exists. And then endpoints are the event or outcome that can be measured objectively to determine whether the intervention being studied is beneficial. So I'm bringing up COVID as an example only because for folks who are reading about it, they're talking about all this stuff in the newspapers. And so, the endpoints in the COVID studies are all, symptomatic infection or asymptomatic infection. So they do a test, a nasal swab, and that is the endpoint of has COVID been detected. But in endpoints in ataxia studies are often things like changes in the SARA rating scale. So that's that rating scale when you go to your doctor, when you do finger to nose, you do this, you stand, you walk, and your doctor will, rate all of that. So that SARA scale is often the endpoint. How is research funded? So the National Institutes of Health, as in your tax dollars, help fund a fair amount of research. The majority of the NIH funding is actually for that basic science research. But as I said, it's that basic science research that ultimately leads to improvements in human health. And when I said the top 10% is funded, what that means is when you submit a grant, only the top 10% is usually funded these days. So 1 in 10 of grants submitted is funded about. Sometimes, depending on the different details, sometimes you get the top 15%. So if of the 100 people submit a grant, only 15 are going to get funded. If it's top 10%, 100 people who submit a grant, only 10 are going to get funded. So it's pretty hard. It's pretty tight. The National Ataxia Foundation also funds a fair amount of research, and the NAF obviously focuses on ataxia research. They are great with a lot of these $50,000, $100,000 pilot studies, which is exactly what we need to galvanize and get the clinical research going, the basic science research going in ataxia. Private philanthropy, of course, also funds a fair amount of research, and pharmaceutical companies also fund a fair amount of ataxia research. And obviously with pharmaceutical companies, there is bias. Ultimately, the company, its goal and its job is to make money. That is the job of the company. I do believe that the individuals at these companies, many of them, I'm not going to say all of them, but many of the individuals at these companies genuinely are trying to improve human health. And I have found that many of the individuals, specifically in the ataxia space with whom I've interacted, they really do want to help. That being said, the job of the company is to make money. So there's a bias there, but there's obviously a common interest, because if it's a good drug that makes money, that means hopefully it's a good drug that improves human health. So there is a balance there, but the pharma companies really are the ones that have the money to do these big clinical trials. Finding that money anywhere else in the process is really hard.
I have a question.
Yes, ma'am.
Talking about money. Let's say, so ataxia medications are in the pipeline, very close at least.
Yep.
If not, they're trying some out right now.
Yeah, I'll talk about it in a bit. Absolutely, they are.
So, but my question is, once they hit the open market, once they prove to be effective, whatever, they go to the open market, are they going to be exorbitantly expensive to purchase, or do you think insurance will cover that?
So yes, my guess is they will be exorbitantly expensive. I think insurance will cover. So for example, the Biohaven research study that I talk about in a bit is trivalolizole. They will get, if it gets approved, if it's found to be safe and effective, and if it gets approved, it will be approved for, SCA 1, 2, 3, maybe 6, 8, and 10, maybe, okay? 6, 7, 8, and 10. So if you have SCA 1, 2, 3, 6, 7, 8, and 10, your physician can probably argue with your insurance company to give you the drug because there's an indication for it, right? So my guess is your insurance company hopefully will relent on that. The problem comes in, I think, for patients who have ataxia of unknown etiology, patients who have SCA 12, just to make it up, right? Those patients, I think, are going to be really difficult to argue with the insurance company, because they're going to say, well, no, it's not approved for your disease. You have SCA 12. It's only approved for SCA 1, 2, 3, 6, 7, 8, and 10. So I think it's going to be hit or miss whether it's approved for ataxias outside of the ones that are specifically indicated, because it is going to be very expensive. That's my guess anyway. And I share your concern. Some of the other ones that I'm talking about in terms of coming down the pipeline, some of those are actually going to be very specific. So it is only a drug for SCA3. So it is, insurance company aside, it is not going to be indicated for any of the other SCAs. So I wouldn't even want to give it to you if you don't have SCA3. So in that regard, I think those will be, those ones you might be able to argue with your insurance company and win if you have SCA3, but medically I wouldn't want to give it to you if you had something else anyway. So there's sort of a, I mean, in that case, it's a medical indication. In the case of trivilazole, it's going to be more difficult because it's a drug. It's pretty, you know, that one's going to be more difficult to argue, but I share your concern about the costs coming down the pipeline. What I would say is this, and I know this isn't going to help anyone on this call, is eventually these patents run out and eventually the prices come down. And so there is that to be said for it, but I realize that we're talking, you know, 10 to 15 years eventually, not right away. And there will be lots of arguments with insurance companies. But certainly I think the opportunity to argue with your insurance company is better than not having that opportunity, if that makes sense. So it's good that they're coming down the market, but I share your concern about the cost. And I don't know what's going to be.
Nancy,
Yes, I see a hand raised. What's up?
Yeah,
Go ahead. Ms. Burkhart. Yep, that's you.
I know, but I forgot.
Okay. Well, if you
love it,
remember,
Nancy.
let me know. That's fine.
If you remember, just chime in.
Yep, absolutely. Not a problem. All right. So I am now going to talk about the COVID vaccine research and myths. And I'm going to include in there some basic science information that will actually become relevant in the ataxia research as well. So I thought I could put it all together. So very big basic science background. Your DNA, your genes, make RNA, which make proteins. And that's the direction the arrow goes. DNA to RNA to proteins. The proteins then do things in our body. Proteins, for example, can attack invading bacteria or viruses. And parts of our immune system called antibodies are proteins. So DNA makes RNA, makes proteins. Proteins are antibodies that attack invaders in our immune system. There are also proteins in our body that actually remove unwanted proteins. So the ataxin 3 protein, what it does is it goes around and it like basically declutters your house, right? So the ataxin 3 protein goes around and declutters things and gets rid of unwanted things, puts bags in Goodwill, sends it out the door. That's what the ataxin 3 protein does. Therefore, when the ataxin 3 protein doesn't work, when the bags that are all bagged up to go to Goodwill aren't able to get out the door, everything clumps together. The proteins, the ataxin 3 proteins, they all clump together. And ultimately that leads to SCA3. So the proteins are the active part that are doing different things, but DNA makes RNA, makes proteins. Proteins do all sorts of things. And you can see proteins can do really good things, but when proteins aren't working right, such as in the SCA3 mutations, when they're not working right, then it can cause problems.
So quick question on that.
Yes.
Just from like a scientific standpoint, the ataxin 3 protein that's not working, is it malformed? Is it missing some elements or?
So what happens is, now good question. So what happens is it's a repeat disease. So when a protein is made, it's a nice floating strand. And it's long, so think of it like a rope. And then once it's made though, it forms a specific conformation.
So this is like the folding.
The folding, exactly.
Okay,
So
yeah.
it folds into a very specific way. And it folds into that specific way, it allows it to bind to things and attach from things and do all sorts of stuff when it's folded in a very specific way. But what happens when there's too many repeats, it basically becomes too long of a rope. And so it gets tangled up and doesn't fold correctly.
Okay.
And when it gets tangled up and doesn't fold correctly, then it can't do all the things it's supposed to do.
Okay.
So we've been hearing a lot about RNA because remember the COVID vaccines, the Pfizer and the Moderna, are mRNA vaccines, messenger RNA vaccines. So what they do, the RNA vaccines, teaches our cells to make antibodies. It teaches our cells to make proteins because that's what RNA does. RNA teaches cells to make proteins. So the mRNA vaccine for COVID teaches our cells to make the antibodies against COVID-19. So that way, when we're exposed to COVID-19 in the world, you know, someone with COVID-19 sneezes on us and we're exposed to it, our body is ready to go. We are ready to fight. We have the antibodies that we need, and we can attack it immediately and eliminate it from our system and not get sick. And the vaccine gives us the blueprint to fight disease. Now, this is what all vaccines do. All vaccines give us the blueprint to fight the disease. The mRNA vaccine just does it in a really fancy kind of cool way because it basically teaches our body how to make the proteins. And it's, as you guys know, incredibly effective. So one of the things I thought was really interesting though, is there's a lot of concerns, and I understand this, about this vaccine being new. And I don't want this new thing, and there hasn't been enough research on it, and it's new, and therefore it's scary. So I was looking up, because I've heard a number of times that like, this is new. So my question was, well, how new is it? So what I actually found, a very abridged mRNA vaccine history, and a lot of this information I actually took from a 2018 article in Nature about mRNA vaccines, and how in 1990, the first mRNA vaccine was used in an animal model, all the way back in 1990, which I actually thought was fascinating. And at the time, though, it wasn't quite ready for humans because there were concerns about the stability of the mRNA, by which they mean the body would see it as foreign and attack it right away before it could do its job of making proteins. So that's what we mean by stability. But it was all the way back in 1990 when it was first used as an RNA model, in an animal model. In 2005, then, Doctors Carrico and Weissman figured out how to make it more stable. And they did this in a very detailed way, swapping out the one agent for, it's very complicated. I won't get into the science of it. They figured out how to make it more stable. And then from 2010 to 2017, there were actually at least 7 different mRNA vaccine clinical trials. Most of those were on HIV. And HIV, for people who know the science of it, HIV is a very different virus than the coronavirus is or anything else. HIV is a much more complicated virus than COVID. So in 2018, there was this article then in Nature talking about mRNA vaccines, a new era in vaccinology, and basically talking about how great these mRNA vaccines were. Of course, by end of 2019, COVID-19 occurred. By January of 2019, they released on the internet the entire sequencing of the COVID-19 virus that was released worldwide for researchers worldwide to be able to do the research on it. And then, of course, by end of 2020, we were able to start vaccinations, and then mass vaccinations in the US and worldwide. And of course, the vaccine has been proven to be very safe and unbelievably effective. And I think in terms of efficacy, I mean, we get the flu shot, and the flu vaccine is anywhere from about 60-ish% effective in a good year. And so this is now, we're talking 95% effective. It's really remarkable, the efficacy of it. But I was amazed, actually, that was back in 1990. And if you guys have time, it's worth going back and reading the story of Dr. Carrico. It is a great story. She came to this country from somewhere in Eastern Europe, I believe, because she had a postdoc at Tufts, then went over to Penn, never was able to get funding, had Dr. Weissman helped her out, but she was always sort of on the bridges. She never was able to get promoted. She had all these issues, but she believed in mRNA and mRNA's potential. And she is now the founder and head of BioNTech. So the Pfizer BioNTech. she's now like the founder and head of it. And so people are basically talking about her and Weissman as Nobel Prize winners, that kind of thing. So it's a great story though about this woman, and she came over with her husband and her kids and didn't have any money. It was in the New York Times in like the late 2020 or so. And there's a bunch of different profiles of her. It's a very interesting story. But anyway, most of this was actually taken from the article in Nature. I do have a few COVID vaccine myths. So vaccines do not have microchip trackers. Vaccines do not alter DNA, because remember, it goes DNA, RNA, protein. So it doesn't go back the other direction. So it doesn't alter that. They do not cause infertility or miscarriages. They do not have terrible side effects. There is a normal range of vaccine side effects have been reported. And of course, the other thing I think is really important to think about is the risk and the benefit. So the risk of the COVID vaccine and the side effects of the COVID vaccine are always less than the side effects of COVID and the risk of COVID. And it's always going to be a risk benefit. Everything we do in life is a risk benefit, right? It is a risk to get in the car and drive because there are car accidents, but we all do it every day. Sure, it's a risk to get the COVID vaccine, but I would argue that the significantly greater risk is not getting the vaccine. Essentially, vaccine side effects are always simply a less severe version of the vaccine symptoms themselves. So anytime you read about some of these things about the vaccine, COVID is way worse. That I know for sure. And then the other myth is I had COVID, so I don't need to get vaccinated. We actually think immunity wanes. And so you actually do need to get vaccinated, even if you had COVID. I can tell you go ahead, please.
So is there really a copy of these slides? There's a lot of people that need to read this.
Robert, we're recording this, and as long as Dr. Rosenthal is okay with it, we will put it up on our YouTube channel.
Cool, great.
Sure, I have to make sure I cited everything correctly, but yes, absolutely.
And our I'll put it on our website.
Yeah, absolutely.
Yeah. There's been some stuff on Facebook last couple weeks about kids that get the Verdura, how it could affect their genes and get ataxia. So it's crazy. Yeah, I know.
It doesn't, crazy. And I, when I was talking with Linda, she said that most of you guys have been vaccinated, but we all know people, myself included, I might add, we all know people who are understandably scared and understandably concerned. And so I think it's a matter of, recognizing the fears are real. The fears are very real and very understandable. But what can we do to sort of weigh the risks and benefits and really think in terms of how can you not only protect yourself, but how can you protect everyone, your friends, your family, your community, et cetera.
Exactly. Yep. The only problem I have with people spreading fear across the multimedia, Facebook, whatever. The people spreading fear.
I agree.
I'm sorry.
So my personal experience, I received the Moderna vaccine in December and January. I had a sore arm after the first dose. I felt kind of crummy after the second dose for about 48 hours, but with some Tylenol, it was okay. My 14 and 15-year-old niece are getting vaccinated this weekend. I'm very excited about that. And I really wish I could get my 6 and 10-year-old vaccinated now. I'm just waiting for the day. And I have been trying to get them signed up actually for clinical trial. I have their names submitted for like every single clinical trial to try to see if I can get them vaccinated. But so far, no one's called me to participate. So I'm hoping I can get in. But I'm certainly, once it is approved for their age range, planning on having my 6 and 10-year-old vaccinated ASAP. That is all I have on COVID and COVID-related things. I was next going to talk about Ataxia clinical research, but I can pause if anyone has any questions or comments. Nope. Okay, perfect.
Keep going.
All right, excellent. So in terms of Ataxia clinical research, there are many observational studies. Many studies are only at 1 or 2 sites. And so I didn't really want to go into all of, at Hopkins, we have a bunch of different observational studies. I'm sure the research near you guys may have some more as well. All over the country, there are these different observational studies at different centers. And so I didn't really want to go into those, but I thought I would talk about the 3 sort of bigger observational studies that are happening. And there probably are more as well, but I'm focusing on the US. And then I'm going to talk about some of the clinical trials as well. But my goal was to focus on some of the bigger ones, as opposed to just the ones that are 1 or 2 centers. So in terms of the CRC-SCA, the Clinical Research Consortium for Spinocerebellar Ataxias, and the purpose of this is to follow individuals with specific spinocerebellar ataxias to understand how the disease changes over time, with the ultimate goal of being able to develop better treatments. Because we need to understand the details of what happens without those treatments in order to then be able to see what happens when people do have treatments. And our other goal is to identify biomarkers of disease progression. And so let me just explain what a biomarker is. You may guys probably have heard this term passed around. It is an objective measurement that may diagnose disease, predict how the disease may change over time, and predict response to therapy. So example, my favorite example is actually glucose or sugar for diabetes. So we can test your sugar, your glucose levels, as a diagnosis of diabetes. So if your fasting glucose, your fasting sugars are above a certain number, you are diagnosed with diabetes. And then similarly, what we can do is we can follow that number over time to see, how you're doing with your therapy. Are you eating healthier? Are you losing weight? Are you taking your medication? Do we need to add more medication? That kind of thing. So if your glucose level, your fasting glucose are going down over time, then that's a good thing. And we are able to monitor your response to therapy. So gee, you've lost 10% of your body weight and now your glucose levels are lower. You responded really well to that therapy. Or we try a medication and your glucose levels haven't changed, then you're not responding well to that medication. So we can follow it over time. And we can also use glucose levels to predict outcomes. So we know people with sky high glucose levels do worse over time. There are more complications, there's kidney disease, there's heart disease, all sorts of stuff. So we can follow it over time. So glucose levels are a biomarker. For the spinocerebellar ataxias these days, we can, if you have one of the genetic SCAs, we can diagnose it with a genetic test. And of course, there are issues with getting that genetic test, as we talked about, but we can diagnose you with a genetic test. What we can't do as well, though, is we can't predict what's going to happen over time. So we know, and if you look around and talk to your friends and people you know, and this group even, with different people with different SCAs, we know that not everyone with SCA1 progresses at the same rate. We know that not everyone with SCA3 progresses at the same rate. Some people have symptoms a very short amount of time, but have a lot of challenges and a lot of problems. Some people can have symptoms for a while and are still doing really well. We know that some of that, who does better and who does worse, is related to the number of repeats you have on the genes. So people who have lots and lots of repeats do worse than people who only have a few repeats more than normal. But what we also know is that those repeats actually only predict about 50 or 60% of the variability. So there's about 40 or 50% of the reasons why there's differences between the people with SCA that we don't know about, and we don't know what they are. And so we want to identify biomarkers to be able to understand and have an objective sense of who's going to do better and who's going to do worse. Does that kind of make sense? So one of the purposes of the CRC-SCA study, the natural history study, you may have heard it called, which I don't like that name, but that's the other name for it. One of the purposes of it is to identify these biomarkers. Anyone ages 6 or older with signs or symptoms of ataxia, and in this case, a diagnosis of 1, 2, 3, 6, 7, 8, or 10. And unfortunately, we don't have the money to test people. So you have to go into the study with a known diagnosis. Unfortunately, we don't have the money to test people. And generally, we don't even have the money to fly people out for their assessments. So unfortunately, for some of these studies, it's hard to participate unless you're able to kind of make your way to a study site. And there's about 17, 18 sites in the country. The IDEA study, the purpose of this study is to develop better methods of, yes, I had a question.
I think that was
Is
just.
that a question or just, all right, that's
Yeah,
fine.
it's not like background noise.
Okay, that's fine. The IDEA study is to develop better methods of measuring movement disorders, really ataxia, by using sensors. So what you're really looking for in the IDEA study is a digital biomarker. And so that's the purpose of it, is we put these little, like, they look like Apple Watches, but these little like opals on hands and on your back and on your chest and on your feet. And you do the same thing that you do in your doctor's visit, but it's all digitized. And so it's much more precise. Because I'm sure you've done this before, where you maybe were sitting in your doctor's office and you heard like the doctor and her fellow discussing whether something is a 1 or a 2. Well, was that movement a 1 or was it a 2? I don't know. What do you think it is? And so there's a lot of subjectivity in this. Whereas the idea of this is a digital biomarker. And so it's all automated. Yes, ma'am.
So I participate in both the SCA study, the CRC, whatever, SCA study, and the IDEA study both.
Okay.
The IDEA study, they actually send you home for 2 weeks. You
Depends
wear
on
those little sensors every day for 2 weeks, and then you mail them back.
Yes, that you know, if you went to University of Chicago, that's the only site doing the where they mail them back. The other sites aren't doing it that way. The other sites were just having them come in every 6 months.
Oh, okay.
Yes, you're right. It was actually, I mean, what's great about the sending home is we all know, and I hear it from my patients all the time, that in clinic, everyone's a little bit better. Everyone's standing a little bit straighter. Everyone's walking a little bit better. There's just something about being in clinic and, you know, you're on and you want to perform. Whereas when you're home, you know, we're all a little more relaxed. And so I think it's great, the home monitoring part of it. Yeah, that's great. And who can participate is confirmed SCA 1, 2, 3, or 6. Also Friedreich's ataxia. And then also siblings with no genetic evidence, or anyone really, controls in general. Doesn't need to be siblings with no genetic evidence of FA or SCA. So generally age matched. And the Friedreich's patients are generally younger. And so we need people who are age matched to the Friedreich's patients and people who are age matched to the SCA patients is the idea. And then the other big observational study going on is the Reddy-Ska study. And the purpose of this is to establish a group of participants with early stage SCA 1 and 3. And then also to determine if specific types of MRI sequences are biomarkers for ataxia. And then also to be clinically trial ready for upcoming clinical trials. And by clinically trial ready, we mean that we have a group of people who are interested in research, who have a known diagnosis, who are early in their disease, and we can therefore follow them over time. And we have a potential biomarker for the research study. So it's called being clinically trial ready. And so that's the purpose of the Reddy-Ska study. You have to have SCA 1 or 3, a genetic diagnosis, and a low SARA score, which in other words, your symptoms can't be too great. And then you can also, though, in this case, be asymptomatic. So if you have a known family history and you get tested, and you do have SCA 1 or 3, you can actually be in the Reddy-Ska as well. So you don't have to have symptoms yet, but you do have to have a known genetic diagnosis. So now, do you have a question?
That study will pay for the genetic testing.
Reddy-Ska will pay for genetic testing, will they?
They'll cover the genetic
testing. At this point, the enrollment's maxed out, yeah.
And you don't need to know the results of it. So you can get tested and not know if you're positive or not.
Correct.
So.
Yeah, because there's a control group as well. So you can get tested, not know your results if you don't want to, but know you're at risk. And there's a control group. So you don't know if you're in the control group or if you're in the pre-symptomatic group, but you can get tested. It's a nice way of doing it. Some people want to know and some people don't. Other comments or questions on these? So I'm going to talk about the pharmaceutical interest in ataxia. And the National Ataxia Foundation established what they call a pre-competitive pharmaceutical consortium. And it's considered pre-competitive because in practice, the pharmaceutical companies are competing with each other. They all want the next big blockbuster drug, right? But they also know that they'll, as a group, they can do better if they pool their resources for a lot of different things. So as I last heard, I believe 10 pharmaceutical companies have signed on, which I translate to be 10 pharmaceutical companies are interested in developing drugs for ataxia. So I like this cartoon. Your Honor, we will prove beyond a reasonable doubt that this pill is both safe and effective. And this is what happens at the start of every clinical trial. We believe the drug will be safe and effective. So there's like a number of different ongoing or coming down the pipeline clinical trials. I talk about like 3 of them, I believe. There's the Biohaven pharmaceutical. They're the ones who actually started out doing clinical trials in ataxia. The drug that they're using is called troriluzole. It's related to the drug riluzole, which is FDA approved for ALS or Lou Gehrig's disease. And there were small studies that showed that riluzole improved the gait and balance in individuals with ataxia. According to Biohaven, troriluzole is better because it will last longer, have better coverage, and is less influenced by what you eat. So you don't have to worry about fasting or not fasting, that kind of thing. And so that's why Biohaven, and there may be other reasons too, but Biohaven will tell you troriluzole is better. So for the troriluzole, they've actually done 2 studies. It's a phase 2/3 study that was only 8 weeks long. So the randomized part, the placebo-controlled part, was only 8 weeks. So for 8 weeks, people were either on placebo or study drug. Then after that 8 weeks was over, participants could elect to continue into what we call the open label part. And in the open label part, everybody knows that they're on drug. So the patients are on the study drug, the doctors know, the researchers know they're on the drug, pharmaceutical company knows they're on the study drug, everyone knows in the open label. But the randomized part was the first 8 weeks. And what they found in those first 8 weeks, after they did all the analysis, it was not definitive, but compelling. And there was a strong placebo response. So we all have a placebo response. So what happened is, the way you look at these things with placebo-controlled studies is how much improved did the placebo group get, and how much improved did the drug get, the group who got the drug get. And we all know in research studies, the placebo group often improves by a little bit. You come in, we're nice, we talk with you, you go home, you're thinking about your ataxia, you exercise more, whatever it may be, we all know that placebo groups improve. So, and the placebo response is really powerful. So the example I always give for a really powerful placebo response is whenever my kids have any kind of gastrointestinal bug and they start throwing up, whenever they start throwing up and I see them throwing up, I get incredibly nauseous. Something about their throwing up makes me feel awful and nauseated and certain that I am going to get sick any minute now. Yes, you agree. No, exactly.
No, I don't think I'm going to get sick. I get sick.
I know. Okay. No, in my case, I think it's all in my head. I think. Because the problem is a couple times I have actually gotten sick because I got the same bug, right? So in my mind, the moment they start throwing up, I'm out. My husband is on hazmat duty, and I am the one who will happily comfort them and hug them once they are cleaned up. Okay, I'm out. So what I found now is I keep Zofran, which is an anti-nausea medication. I have now got to the point where I will keep it in the house. Because what happens is they will throw up. If I take a Zofran, I will feel better within 2 or 3 minutes. And here's the thing, I know intellectually, based on my medical school and science background, in the 3 minutes since I have taken this drug, it has had 0 opportunity to get to my brain to make me feel less nauseous. And it is all in my head. It is 100% all in my head, but I'm telling you, it feels so real and it works. and every single time. So that is my own personal experience with placebo, right? So the placebo effect is really powerful. So what happened in this initial study is the people on placebo got a lot better, and the people on drug got a little bit more better than the people on placebo, but it wasn't adequately powered because the difference between placebo and drug wasn't high enough. So now there's an ongoing phase 3 study. It's about 42-ish week long, give or take, and it is a randomized controlled 42-week long study on a higher dose of the drug, where people are getting a placebo or drug for 42-ish, maybe 48-ish weeks. I forgot which one, right around there. And the idea is it's placebo controlled. And then afterward, people switch over to open label. The original plan was to complete enrollment for that phase 3 study. Enrollment probably was supposed to end by about summer of 2020, but basically COVID put a 1-year hold on that. And so enrollment actually ended in January of 2021. It's about a 6-7 month delay because of COVID. Because essentially when COVID hit, at every single site across the US, we were not allowed to enroll any, research stopped basically. And so that study is ongoing. Hopefully we'll have the results soon. So that is the Biohaven. It is a pill. It is twice a day. There is also coming down the pipeline a gene therapy trial for SCA3. It will be administered cerebrospinal fluid, so it's called an intrathecal injection, meaning it goes into the spinal fluid one time, and that's it for 5 years. And the idea is this gene therapy, it replaces the SCA3 mutated gene with a non-mutated gene replacement. And it uses what's called a viral vector, which is the same type of concept as the AstraZeneca COVID vaccine. It's a viral vector. It's a different type of viral vector, but it's that same idea. So this trial is coming down the pipeline and will be starting by when it'll start the lead-in. It's going to be a long lead-in before anyone's actually getting the gene therapy. It's going to be starting in about winter of 2022. So we got some time, but it's coming down the pipeline, gene therapy. They're starting with SCA3, but I have every intention, every expectation that what they do on 3, they're going to be doing relatively soon on 2 and 1.
So I'm kind of a
And
science.
the reason they're doing 3, 2, and 1 is those are the most common. What's that?
I'm kind of a science geek. I have a question. Is this
Please.
Gene therapy using CRISPR?
No, I don't think so.
Okay.
Because it's not CRISPR. It's a virus vector, and so the virus will insert the gene in and take out the other one. I don't think it's a CRISPR related necessarily.
Okay,
It's a good question. It's still very cool. So this is coming down the pipeline. There is another trial coming down the pipeline. And this is an antisense oligonucleotide. So I know we've talked about this. Remember, DNA makes RNA, makes proteins. And what the ASOs, the antisense oligonucleotides, it binds to the RNA. So it therefore stops the RNA from making the bad protein. Because even though I know we've talked about, DNA makes RNA, makes proteins, it's an ongoing process. So it's not like once the gene makes the protein once, that's it, it's done. It keeps making the protein. So the DNA, the mutated DNA keeps making the mutated RNA, which keeps making the bad protein. And the idea of the ASOs is by binding to the RNA, it stops production of the bad protein. There are this technology, this ASO technology has actually already been used for one particular disease that can be devastating in children, and now the kids are actually living and doing well. It is coming soon, probably starting with SCA3. And I like this trial, this cartoon. It says, this really is an innovative approach, but I'm afraid we can't consider it. It's never been done before. So I thought that was a nice a catch-22.
I do have a question.
Yeah, please.
Okay, so it binds it from making bad protein, but is there then the good protein in the system, or do they have to supplement that?
So they're not supplementing it, because the idea is that we know there's a couple of different reasons why we think we can get away with this. we know that there's within nature and within our bodies, there's oftentimes lots of pathways that do overlap. So in people with SCA3, the regular normal SCA3 exists. So because the regular SCA protein doesn't exist, only the mutated one exists, it's okay if we stop only the mutated one. And that clearance of protein earlier, that can happen with lots of other proteins. So there are lots of, in other words, there's lots of people in the house who package up everything and give it to Goodwill. So if the SCA3 protein isn't working, there's lots of other proteins that can do that same job. So they don't have to necessarily give the good protein. Sure, it might help if they did, but you don't necessarily have to. If your goal is to slow or stop progression, you don't necessarily have to. And it might be that the ASOs, let's say they slow progression by only 50%. Okay, sounds great. Let's start, anything is better than nothing. And the hope is that it can do even more than that. I know there's some other clinical trials that are also coming down the pipeline, and I wasn't sure what I was and was not allowed to talk about. I know there's a study looking at baclofen and clozoxazone coming down the pipeline. I don't know if that's gotten funding, but there's other things that are coming down the pipeline as well, but I never know what I am and I'm not allowed to talk about. But in summary, basic science and clinical research are needed to find better treatments and ultimately cures for ataxia. The COVID vaccine went through the same research process as every other vaccine and every other molecule you've ever taken from your doctor. But the idea is they got rid of a lot of the red tape, and more clinical trials for ataxia are starting soon. Oh, and then I have one final cartoon. So I like this. It says, I don't have any accurate numbers, so I just made up this one. And the dollar amount is like 4,629,000. And then he says, studies have shown that accurate numbers aren't any more useful than the ones you make up. And the question is, how many studies show that? And its answer is 87. Not like that cartoon. Yeah. And I'm happy to take more questions.
Well, they say that 90% of stats are made up, so.
I don't know if you quite saw the humor, but I was emailing a biostatistician and I had my subject line being lies, damn lies, ... I don't know if you quite saw the humor, the quote, lies, damn lies, and statistics. Yeah, I never quite saw the humor, but I see a question. Yes, ma'am.
I thought of my question from before.
Please.
When you talk about new drugs being developed for ataxia, is that to cure the disease or is it to lessen symptoms?
Both. So the genetic treatment, if you cure it, you'll lessen symptoms. So that's number one.
Wow,
but there are also, there are also studies where the purpose is symptomatic management. And I know that there's the baclofen clozoxazone combination study that I don't have a slide on. What the purpose of that is both symptomatic management and maybe disease modification. Originally, the Biohaven study, the reason it was only 8 weeks originally is they were aiming for symptomatic management and helping symptoms. And what they found is that it may actually slow the course of the disease. There's a lot of studies looking at exercise for symptomatic management of ataxia, showing that it works basically, and that it improves your symptoms now. So exercise is one that always comes up as sort of a current treatment that you can do now. And then otherwise, a lot of like the original, if you go to your doctor and you talk with him or her about the different medications you can try, all of those are based on small studies of symptomatic management. So Prozac and amantadine and Buspar and Riluzole, all of those were small studies showing improved symptoms in people with ataxia. But the ASOs and the other gene therapy trial, the purpose of that is to really slow to stop the disease. That's the purpose of that.
Thank you.
You're welcome. Other questions? Yeah, I can stop sharing.
I found your talk very, very interesting and informative, and it's exciting
Very clear.
to
Very
be
clear.
on. It's exciting to be on the, having ataxia and thinking, oh, there might be something to help me soon.
I agree. I don't know. It's such a slow process.
It
And
is.
let's just, for example, if you take the gene therapy trial, it's starting, hopefully by 2022, 2023, which then means 5 years later, we're still at, 2028. Presumably in that time, they're gonna, if safety holds out, they're doing it in 3 right now. I hope if the safety holds, they'll then try it in 2 and 1. I don't know, or 1, probably in that order, 2 then 1, or maybe 1 then 2, we'll say. And so, but either way, we're not talking, by next year. But there's definitely things coming down the pipeline. I just wish it were faster. Are you on mute?
I'm okay with delayed gratification, particularly since I literally thought there'd be nothing in my lifetime. So even if there's something for me when I'm 60, it's still better than what I thought.
No, my opponents thought that there would be a cure for ataxia in patient. And I had a patient ask me, how exactly is she defining generation? Was the question. And I don't know, Dr. Perlman's, that was what Perlman said, that there will be a cure in this generation. So there's no doubt, I think a couple pharmaceutical companies, and that's actually huge and really important. And with that, I think will come more opportunities and even more interest. It's just slow. That's the problem. Yeah.
I have a
Other
question.
questions or things I can
Yeah,
help
I
with?
do.
Yes, sir.
If the bad protein kills cells in your brain, can the cells be regenerated with a good protein? Or?
No, generally not. No, not in the brain anyway. Generally not. But let me say this though. What we know the brain can do is it's pretty good at building other pathways. So not perfect, not great. That's not, but so let's say I had magical pixie dust and I could sprinkle it on all of you and stop your ataxia exactly where it is, okay? Magical pixie dust, which I wish I owned, but I do not. Magical pixie dust. Different folks will say they have different challenges from their ataxia at this point. And what's always amazing though, is over time, the brain can build these other pathways that aren't the same as, but help. And the example I always give is that, if there's a gigantic pileup on the Beltway, 18-car crash, the Beltway stopped in all directions, it's awful, no one can get through, which means that everyone is taking these side lanes, the little 1 and 2 lane roads, and traffic is awful, right? So if you build up those 1 or 2 lane roads to being 2 or 3 lane roads, it's still not the same as the Beltway. The Beltway was always faster. The Beltway was always a little bit better. But if you build up those 1 and 2 lane roads to become 2 or 3 lane roads, you're still doing better than you were before. So that's kind of the analogy. And it's, you can, even now, even without my magical pixie dust, there are things that you can do to take those 1 and 2 lane roads and build them up a little bit more to the 2 or 3 lane roads. And we are actually, I know at least at Hopkins, I know there's a couple of different research studies going on to try to figure out better ways of doing that. And can we, stimulate, do different types of stimulation to the cerebellum? And if we stimulate the cerebellum at the same time that we do exercise, can we help build up those, 1 and 2 lane roads to become 2 and 3. Not the same as the Beltway, but better than it was before. And those are things that you can do now even. Question?
No, just comment. So I know exercise helps ataxia, but I really wish sitting on your couch and eating cookies was helpful to ataxia, because that is more my.
Well,
So I literally said to him, I'm gonna work out. Like today, this afternoon, I'm like, I'm gonna exercise because I know it helps ataxia. And he's like laughing at me. I'm like, because I'm wishing that just sitting on my couch and did nothing.
Linda, I wish sitting on my couch and eating cookies would allow me to lose my COVID weight and get fit back into my clothes and be heart healthy and, decrease my risk of high blood pressure and cholesterol and all those things. I'm right there with you. I would much rather sit on my couch and eat cookies. But that being said, at least over here for me, it is a beautiful 70-something degree day outside. And a bike ride will do me some good, which is my plan for this afternoon, because it has to be done for all of us. For those with ataxia, even more so. But yes, I will be putting on my workout clothes and going on a bike ride because it simply has to be done. It is a beautiful day outside, at least for me over here. And so I can't not take full advantage of that. But yes, I'm totally with you. I wish sitting on my couch and eating cookies would make me healthy, but it doesn't. Yeah.
Thank you.
You're
But
welcome.
we can so try work and Sonic out to eat cookies. I do a morning walk every morning. I go up for about up to 2.5 miles walk. I
Great.
call it my drunk walk because I use the whole sidewalk when I walk.
You're getting out there and doing it. And sometimes in my neighborhood, if I'm like biking or walking or something, I'll see, folks who are very clearly like, old enough to be my grandparents, like still out walking and running. And it's that reminder of like, no, that needs to be me. And so that's what I need to do. But yeah, it's hard. You got to get up every morning and do it, myself included. And I
hate when it passes by. I'm like, slow down. Sorry.
I need my exercise rings to close at least, 5 days a week. That's my goal on my watch. They have to close at least 5 days a week. So, anything else? Any other questions?
Nope. Thank you very much.
Thank you.
Thank you.
All right,
We appreciate it.
Thank you for having me.
Thank you for
having me.
making it so clear for to understand better.
Well, it was my pleasure. And I'm if anyone has any follow-up questions, Linda knows how to get in touch with me.
Sounds good. Thank you very much, Doctor.
All right. Well, I guess I will sign off and I will see you guys all. So hopefully at the, in-person NIF meeting in Orlando in 2022. That's the goal.
Absolutely.
All right. I'll see y'all then. Thank you.
Bye.
Thank you.
Bye.
Have a good weekend.
Okay. Thank you for having me. Bye. You too.